15% of women still had enough of the drug in their blood to impair their driving 8 hours after taking a common sleeping pill. For men, it was 3%. Same medication, same dose, 5 times the risk. The drug was zolpidem, better known as Ambien. In 2011, 39 million prescriptions for zolpidem products were dispensed in the United States, and nearly 2/3 of the patients were female. So how did such a large difference remain hidden for such a long time? Because for most of modern medical history, the male body was treated as the standard human body. Women were often excluded from research because their hormones were considered too complicated. Menstrual cycles might make the data less predictable, and pregnancy might create risk. So researchers chose what seemed like the simpler option: study men, then assume the results apply to everyone. But removing women from studies did not remove the biological complexities. It removed the evidence needed to understand it. Clinical trials are supposed to answer a basic question: what does this treatment do inside the human body? But there is no single human body. People differ in age, weight, genetics, anatomy, health conditions, hormone levels, and lifestyle. Every difference can make the data harder to interpret. Researchers often try to reduce that variation. The more similar the participants are, the easier it is to see what that drug is actually doing. For decades, women were treated as a source of unnecessary variation. Hormones can change during the menstrual cycle. Pregnancy can transform the body, and menopause creates another set of biological changes. Instead of studying those differences, researchers often removed them from the experiment. They mainly studied on male animals and male participants and extrapolated the results onto women. They tested treatments mainly on male animals and male participants and then extrapolated the results onto women. The male body became the default, the female body became a complication. This affected more than drug trials. Women were also underrepresented in research into major diseases, including heart disease, even though at least 500,000 women were dying from it each year. The same problem appeared in drug safety. Between 2000 and 2003, 4 out of 10 prescription drugs withdrawn from the market caused fatal heart rhythm disturbances more often in women than in men. When women are excluded from the research, these patterns can remain invisible. The same problem appeared in drug safety. Between 2000 and 2003, 4 out of 10 prescription drugs withdrawn from the market caused fatal heart rhythm disturbances more often in women than in men. When women are excluded from the research, these patterns can remain invisible. The experiment does become easier to manage, but the treatment also becomes harder to trust. One of the clearest examples of this began with a drug called diethylstilbestrol, usually shortened to DES. DES was a powerful synthetic form of estrogen. Starting in 1947, it was prescribed during pregnancy because doctors believed it would prevent miscarriage. Worldwide, an estimated 10 million people were exposed to it, including 4.8 million pregnant women in the United States. But the drug did not work. Rigorous trials in the 1950s found that DES was ineffective in preventing miscarriages. That should have ended the story. But instead, it continued to be prescribed for roughly another 2 decades. Pharmaceutical companies promoted it, physicians saw other physicians using it, and once the treatment becomes common, familiarity can begin to feel like proof. And then the consequences appeared. DES could cross the placenta and reach the developing fetus. It was eventually recognized as both a carcinogen and a teratogen, meaning it could cause cancer and disrupt fetal development. Some daughters exposed before birth developed abnormalities in their reproductive tract. Others developed vaginal clear cell adenocarcinoma, an extremely rare cancer. The cancer affected fewer than 1 in every 1,000 exposed daughter, but across millions of exposures, even a very rare outcome matters. DES, together with thalidomide, which was a drug prescribed in the late 1950s and early 1960s for nausea during pregnancy and caused severe birth defects in thousands of babies, forced scientists and regulators to recognize how vulnerable fetal development can actually be. But the lesson medicine took from these 2 disasters created a different problem. Instead of improving research involving pregnancy, the system often avoided it entirely. In 1977, the United States Food and Drug Administration recommended excluding women of childbearing potential from early-stage drug trials. That was the language used at the time. The reasoning was understandable. If a participant became pregnant during a study, the experimental drug could hurt the fetus. But childbearing potential was an extremely broad category. It could include almost any woman from puberty to menopause, regardless of whether pregnancy was realistic. A policy intended to prevent harm removed a large part of the population from medical research. Researchers learned less about female bodies while the same drugs were still prescribed to women after approval. The danger had not disappeared. It had simply moved from controlled research to ordinary medical care. The problem also continued after more women began entering trials. A 2018 study of 107 NIH-approved clinical trials shows that 72% of them performed no sex-specific analyses. If you're interested in joining a community of like-minded people who are also interested in learning about the little things that we ignore in our daily lives, join our Telegram channel. We also share additional resources there on each of the topics that we cover. Women were technically involved, but the results were still being combined into one average. Inclusion without analysis can hide the very differences that inclusion was supposed to reveal. By the late 20th century, women's health advocates, physicians, and scientists were pushing back. A major turning point came with the NIH Revitalization Act of 1993. The law required women and minorities to be included in research funded by the National Institutes of Health. More importantly, trials had to be designed to identify whether results differed between men and women. Then in 1998, the FDA required new drug applications to present safety and effectiveness data separately for men and women. A drug might work well with men, but cause more side effects in women. Combine everyone into one average, and that pattern can start to disappear. This is the principle behind treating sex as a biological variable. Sex can influence how a disease develops, how symptoms appear, how a drug moves through the body, and how quickly it is removed. It is not a distraction from the science, it is part of the science. After you swallow a pill, it begins a journey in your body. It has to be absorbed, distributed through the body, metabolized, and finally excreted. And sex can influence every part of this journey. Differences in body fat, weight, and digestion can determine how widely a drug spreads throughout the body and how quickly it will reach the bloodstream. The menstrual cycle, menopause, and pregnancy, they can all influence how a medicine behaves in the body. During pregnancy, estrogen levels can rise around 30-fold by childbirth, and albumin, a protein that binds to many drugs in the blood, can fall to 70 to 80% of its normal levels. These changes can alter how much medication remains active in the body. Liver enzymes may also differ. An important enzyme called CYP3A4 has been reported roughly twice the level in women. That does not mean that women always need lower doses. For some drugs, faster metabolism can reduce the effectiveness. For others, slow clearance might increase its side effects. The point is that not every difference produces the same result. Researchers need to test the difference instead of assuming that it doesn't matter. Zolpidem shows what happens when that difference is finally measured. Women on average clear the drug from their bodies more slowly than men. After a standard 10 mg dose, 15% of women still had blood concentrations high enough to impair driving 8 hours later. That number was 3% for men. With the extended release version, 33% of women and 25% of men still had potentially impairing levels after 8 hours. A person can go to sleep, wake up, believe the medicine had worn off, get into a car, and still be significantly impaired. The FDA eventually required lower recommended doses for women. The medicine had not changed, the female biology hadn't changed. What had changed was that regulators had finally realized that they needed to look at the data separately. The danger had been there all along, but the average had hidden it. Pregnancy remains one of the hardest areas in medical research. People who are pregnant have often been described as a vulnerable population. That sounds protective, but pregnant people still develop infections, live with chronic diseases, need vaccines, and require treatment. When they're excluded from research, clinicians are forced to make decisions with little reliable evidence. Fewer than 20 FDA-approved drugs are specifically approved for use during pregnancy. Yet many other drugs are still used during pregnancy without having been adequately studied for that purpose. On average, it takes 27 years after a prescription drug has been approved to determine its potential to cause birth defects. 27 years. An entire generation could be exposed before the risks become clear. Including pregnant people in research can create risk, but excluding them also creates risk. It transfers uncertainty from a carefully monitored trial into everyday medical care. This is why many experts argue that pregnant people should be treated as a special population instead of being automatically excluded as a vulnerable one. Stronger safeguards might be necessary, but protection shouldn't automatically mean exclusion. For decades, medicine treated women's bodies as a complication. Hormones made the data too messy, pregnancy made the research too risky, and sex differences made the data harder to summarize. So the system often chose the easier path. It studied men and called the results universal, but the complexity never really disappeared. It returned later as unexpected side effects, dangerous doses, ineffective treatments, and gaps in medical knowledge. Even today, including women is not enough. Researchers need to examine what happens to them. The lesson isn't that every man and every woman responds differently to every drug. Human biology does not fit into two perfectly separate boxes. Sex, hormones, body composition, age, pregnancy status, genetics, and many other factors interact. The lesson is that variation matters. A clinical study should not be based on the easiest body to study. It should be designed around the real people that will eventually receive the treatment. Because excluding someone from research does not eliminate the risk that they face. It eliminates the knowledge needed to protect them. And in medicine, what we choose not to study can be just as dangerous as what we do not know. In our previous video, we talked about how cities and public spaces exclude anyone who falls outside of the standard. So if you want to see how the same patterns appears in urban planning, watch that video next.